Good morning students. In the last video we discussed about basic structure of eukaryotic cell. We also discussed about cell membrane, its composition, structure of cell membrane or plasma membrane. We also discussed some of the models which was given to propose to explain the structure of plasma membrane. First one we studied the Robertson model. Next we studied about Davison and Daniel model, that is also termed as sandwich model. But both of the models are rejected because they fails to explain the selective permeability of plasma lemma. Then later on one more model was proposed, that is Flute Mosaic model. It was given by Singer and Nicholson, and its latest and most widely accepted model for the structure of plasma lemma or plasma membrane. According to Flute Mosaic model, proteins are arranged in phospholipid bilayer as mosaic pattern. Thus membrane is termed as protein iceberg in the sea of phospholipid. Phospholipid का sea और उसके बीच में प्रोटीन सरफेस पे मिडिल में प्रेजेंट है. This is also known as गुलाब जामुन in a concentrated solution of sugar. गुलाब जामुन को कंपेयर किया गया प्रोटीन से और कंसनट्रेटेड सॉल्यूशन है वो माना गया है फॉस्फोलिपिड का. लेट्स नाउ डिस्कस कि हमने इस मॉडल को फ्लूइड मोज़ाइक मॉडल क्यों बोला. क्योंकि जो फ्लूइड नेचर है it comes from fluidity. Due to the fluidity of the membrane इसको फ्लूइड बोला गया. जो लिपिड मॉलिक्यूल्स हैं एक्चुअली they are linked with each other by very weak bond. Okay, this bond is very weak. Hence it allows lateral movement. That's why we termed it as flute. Now why it is termed as mosaic? Because we have already discussed in the last video that proteins are present, some proteins are present on the surface, some are buried in the membrane, some are fully buried, some are partially buried. So they are arranged in a random fashion or random distribution. It looks like a mosaic pattern. That's why this model is termed as flute mosaic model. It was given by famous scientists Singer and Nicholson in 1972. Hence, due to the fluidity of the membrane, fluidity means to flow, it termed as fluid, and lipids can move, whereas protein can also move, but not to this much extent to which the lipids can move. Okay, that's why we termed it as a fluid mosaic model. I also said that it's protein iceberg in the sea of phospholipid. This is how the exact structure of plasma membrane looks like. See this red portion, it is termed as hydrophilic head. This central portion, it is hydrophobic tail, and this is the central region. Blue color structure, this one, this, these are actually proteins. There are two types of proteins, as I have already told you. The protein which are present on the surface, they are termed as peripheral protein or surface protein, and the proteins which are present inside, like this is fully buried, and these are partially buried. So these blue color structures are actually protein, and other than protein and lipids, small amount of carbohydrates are also present. Here you can see that glycolipid, glycoprotein, these are also present. And this yellow color structure, this one and this one, these are actually cholesterol. Here you can see this yellow color structure is termed as cholesterol. This is integral membrane proteins. These are fully buried, this is partially buried. Peripheral protein present on the surface of membrane. These are protein channels which will allow the movement of molecule. We will study about this in the transportation through plasma membrane. Now we will discuss about composition of plasma membrane. We have already discussed about it in the last video, but we will take a quick review of it. We will discuss in the following heading: phospholipid and proteins, because it's mainly formed of lipid and protein. Now we will talk about phospholipid. These are actually main component of cell membrane. What are phospholipids? These are main components of cell membrane or plasma membrane, because it forms continuous structural frame of cell membrane. It forms continuous structural frame of cell membrane. In this, the lipids are arranged within the membrane with the polar head towards the outer side. We have discussed about it. This polar head, also known as hydrophilic head, and these bilayer hydrophobic tail. So in it, the lipids are arranged within the membrane with polar head towards the outer side and the hydrophobic tails towards the inner part. This ensures that non-polar tail of saturated and unsaturated hydrocarbon, it's completely protected from the aqueous environment, and obviously it's hydrophobic, yani water hating part, so it is protected from aqueous environment. This phospholipid layer, lipid layer actually provide fluidity. We have discussed earlier, it provides fluidity to the plasma membrane because phospholipids are rich in unsaturated fatty acid which are liquid in nature, and this nature of liquid enables the lateral movement of protein within bilayer, within the overall bilayer. This fluidity nature of membrane allows the movement of protein within overall bilayer, and that's why we said that the fluid nature or the fluidity. Now, what is the function or what is the advantage of this fluid nature of membrane? Actually, this fluid nature of membrane, it helps in very important function like cell growth, it also helps in formation of intercellular junctions between the adjacent cell, formation of intercellular junction. It also helps in endocytosis. We will discuss these terms later on. Endocytosis also help in cell division. Hence, this is all about phospholipid. Now we discuss about protein. We have already discussed about phospholipids. Now we are discussing about proteins. As we have already discussed that on the basis of ease of extraction, there are two types of protein: integral protein and peripheral protein. The integral proteins, also known as intrinsic protein, and these proteins are further of two types. One is partially buried and other one is totally or fully buried. Some integral proteins which are totally buried through the complete thickness of membrane, they are present. As we have discussed when we talk about the structure of plasma membrane through the help of a diagram, we studied there that integral proteins are totally buried through the complete thickness of membrane. These type of proteins are also known as tunnel protein. Proteins which are totally buried are also known as tunnel protein or channel protein, which provides the passage for movement of water soluble material across the membrane. Now peripheral or extrinsic protein. As the name suggests, extrinsic or peripheral, they are arranged outside and they can easily be separated. These proteins have enzymatic activity, and there is an helical type of extrinsic protein which is known as spectrin. It's helical type of protein. Helical type of extrinsic protein. It is found on the cytosolic phase, यानी the phase which faces the cytoplasm. इंटरसेलुलर स्पेस जो है उसको जो फेस करता हुआ, साइटोप्लाज्म को फेस करता हुआ स्पेस है, वहाँ पे प्रेजेंट होता है. और जैसे ये आपकी मेम्ब्रेन है, मैं भी आपको डायग्राम दिखाऊंगी. ये मेम्ब्रेन है, यहाँ पे हेलिक्स की फॉर्म में प्रेजेंट है और इधर क्या प्रेजेंट है, साइटोप्लाज्म. और इस प्रोटीन का नाम है स्पेक्ट्रिन. और स्पेक्ट्रिन जो है ये साइटोस्केलेटन का पार्ट है. अब साइटोस्केलेटन के बारे में हमने डिस्कस किया था, यानी बेसिक फ्रेमवर्क प्रोवाइड करो. ओलिगोसैकेराइड्स ऑफ अ ग्लाइकोलिपिड एंड ग्लाइकोप्रोटीन्स आर प्रेजेंट ऑन द आउटर सरफेस of a plasma membrane, and they are involved in cell to cell recognition mechanism. What actually function they do? They are involved in cell to cell recognition mechanism. Plasma membrane is actually a asymmetric structure because carbohydrates are present on the outer surface, and spectrin protein, it is present here like in this manner. This is spectrin. The spectrin protein, it is present on the cytoplasmic phase of the membrane. According to this theory, phospholipid molecule are found in semi-fluid phase. They are found in semi-fluid phase, and the intrinsic protein exhibit a flip-flop movement. This intrinsic proteins, they exhibit flip-flop movements, and protein layers provide elasticity to the plasma membrane, and the phospholipid molecules provide selective semi-permeability to the membrane. So this is all about fluid mosaic theory for the structure of plasma membrane. Let's now discuss the functions of plasma membrane. It provides shape to the cell. It's the end boundary, so definitely it will provide the shape to the cell. It protects the cell organelle. ये एक बाउंड्री की फॉर्म में इसके अंदर सेल ऑर्गेनल्स प्रेजेंट है, तो उसका प्रोटेक्शन का काम भी ये करेगा. इट हेल्प्स इन सेल एडहेशन, सेल एडहेशन में भी हेल्प करेगा. इट ऑल्सो हेल्प्स इन द ट्रांसपोर्टेशन ऑफ सब्सटेंसेस. ये एक इसका बहुत ही इम्पॉर्टेंट फीचर है जिसे हम अभी डिटेल में पढ़ने वाले हैं. इट इज़ ऑफ टू टाइप्स, पैसिव ट्रांसपोर्ट एंड एक्टिव ट्रांसपोर्ट. देखिए जो पैसिव ट्रांसपोर्ट है, इट इन्वॉल्व्स द मूवमेंट ऑफ सब्सटेंसेस फ्रॉम देयर हायर कंसंट्रेशन टू लो कंसंट्रेशन. दैट इज, it's the movement of molecules along the concentration gradient. Its example are diffusion and osmosis. Diffusion and osmosis are the example of passive transport. Whereas the active transport, it involves the movement of substances against the concentration gradient. यानी अब जो movement होगी वो होगी their low concentration to their higher concentration. It involves the use of ATP or the energy currency because it's the movement against the concentration gradient, so obviously it will require an energy, and energy will be in the form of ATP. In this process, carrier protein act as a carrier. I have already told you earlier that carrier protein, channel protein or tunnel protein, these proteins helps in the transportation of molecules. So this active transport occur with the help of this carrier proteins. Now we will discuss about passive transport in detail. Two common examples of passive transport is diffusion and osmosis. And diffusion is also further of two types. Diffusion is actually the movement of molecules from the region of higher concentration to the region of the low concentration. It is of two types, simple and facilitated diffusion. There is two type of diffusion. We are discussing this with the help of a diagram. So here you can see I have shown two cells. In this cell, first one, here the outside concentration is high and inside concentration is low. और यहाँ पे जो मूवमेंट हो रही है वो कहाँ से हो रही है, हाई कंसंट्रेशन से लो कंसंट्रेशन की तरफ. And in second cell, see, outside the concentration is low whereas inside the cell concentration is high. So यहाँ पे मूवमेंट कैसे होगी, हाई कंसंट्रेशन से लो, यानी कि इनसाइड टू द आउटसाइड. So this is all about simple diffusion. Now we will study about facilitated diffusion. As the name suggests, facilitated, यानी something is needed to facilitate or to ease the transport. So as the polar molecules cannot pass through non-polar lipid layer, they require carrier protein. उनको carrier protein की ज़रूरत होती है to facilitate their transport across the membrane. So see here, this is carrier protein and it's carrying the substances or the molecules to be transported from outside to inside the cell, and this is transported. See, now it is finally released. So this is all about facilitated diffusion. Transport proteins, they helps the particle to move across the membrane. We will discuss about active transport. What happens in active transport? It is the movement of molecules from their low concentration to the higher concentration, or we can say that it is against the concentration gradient. Against concentration gradient. For this carrier proteins of cell membrane, इसके लिए जो carrier proteins होंगे cell membrane पे वो act करेंगे as pump. कौन act करेगा pump की तरह? Carrier protein. And this will allow the movement of molecules. ATP is needed because we are working against the concentration gradient. That's why energy is required. Energy currency is in the form of ATP, and from where ATP is produced? ATP is produced through cellular respiration in every cells. तो होगा क्या? Molecules, they will attach itself to carrier protein once, and when the particle is released, the carrier protein come back to its original shape. We will study with the help of a picture. So here you can see a molecule attach itself to the protein pump, and ATP is converted into ADP plus inorganic phosphate, यानी कि एनर्जी का यूज़ हो रहा है. इसको हम बोलेंगे एक्टिव ट्रांसपोर्ट. ये देखिए क्या होगा, ये जो मॉलिक्यूल है ये रिलीज़ हो जाएगा इनसाइड द सेल. तो हैंस पंपिंग ऑफ मॉलिक्यूल्स अक्रॉस द सेल मेम्ब्रेन यूजिंग अ प्रोटीन पंप, इट इज़ टर्म्ड एज एक्टिव ट्रांसपोर्ट. can see difference between passive transport and active transport. We studied passive transport. It is of two types, diffusion and facilitated diffusion. Diffusion occurs from region of high concentration to region of low concentration. In facilitated diffusion, it also occurs from high to low, but it needs channel or carrier proteins for the transportation of molecules across the membrane. Whereas in active transport, it needs protein channels or carrier protein, and it also requires the ATP, and the transportation occur from the low concentration to the region of their higher concentration. Next point is it performs endocytosis. Now what is this endocytosis? The intake of substances with the help of plasma membrane is termed as endocytosis. जो intake हो रहा है substances का प्लाज्मा मेम्ब्रेन के अक्रॉस उसको बोलते हैं एंडोसाइटोसिस. अब जो ये एंडोसाइटोसिस होता है ये टू टाइप्स का होता है, फैगोसाइटोसिस एंड pinocytosis. As the name suggests, phagocytosis, it is the intake of solid substances, whereas pinocytosis, it is the intake of liquid substances. Here you can see phagocytosis. It is also known as cell eating. Here is a solid particle which need to be ingested or engulfed in. So ingestion of solid complex material by the membrane in the form of vesicle. Here the name of vesicle is phagosome, is termed as phagocytosis. For this, membrane produces a pseudopodium. As you know, we have read in amoeba, it has pseudopodia like structure, and it produces pseudopodia and take the food material inside it. So here pseudopodium is produced to take this solid particle inside the membrane. Okay, now next is pinocytosis. It is also known as cell drinking. Okay, इसमें क्या होगा, ingestion of liquid material. As you can see, here is the liquid material present. Ingestion of this liquid material by plasma lemma or by plasma membrane in the form of vesicles or bag like structure. This vesicle is known as pinosome. This is termed as pinocytosis. The next function is it performs exocytosis. Exo, as the name suggests, expulsion of waste product from the cell. Endocytosis means the intake of substances with the help of plasma membrane. Intake of substances. In exocytosis, what is done? Expulsion of waste product. It is also known as cell vomiting. Egestion of waste material or the expulsion of waste material from cell through plasma membrane is termed as exocytosis. Okay, we will study some of the modifications of plasma membrane. In animal cell, the plasma membrane forms certain structures to perform special function. General or common functions we have already studied. Now we are studying some special functions, that are the epithelial cells form finger-like processes called microvilli. Like this is, assume this is epithelial cell layer. They have these finger-like structures present, finger-like processes. These are termed as microvilli. Now what is the function of microvilli? They increase the surface area for absorption. सरफेस एरिया इंक्रीज़ कर देंगी एब्जॉर्प्शन के लिए, ट्रांसपोर्टेशन इंक्रीज़ करेंगी, सेल सिक्रीशन में हेल्प करती हैं, सेल एडहेशन में हेल्प करेंगी, सेल एब्जॉर्प्शन में हेल्प करेंगी. It forms desmosomes. Now what are these desmosomes? Actually the adjacent cell, assume these are two adjacent cells, these are connected with intercellular junctions. These are termed as desmosomes that will provide strong adhesion between the cells. These two cells are very strongly adhere to each other with the help of these desmosomes. So what it actually do? It will provide strong adhesion between cells because they are linked intercellularly to the intermediate. It forms desmosomes between adjacent cells which provide mechanical strength and help in cell-to-cell adhesion. Now what are these desmosomes? These desmosomes are actually intercellular junction. Assume that this is a cell. this is another cell. Yani these are two adjacent cells. They are connected with each other with the help of these desmosomes. These desmosomes are intercellular junction which will provide strong adhesion between cells. दो cells के बीच में एक बहुत strong adhesion provide कराएंगी. And one of the main function, as I have already told, that it gives mechanical strength. ये एक दूसरे से बहुत strongly bonded होंगे कि ये tissues को क्या करेंगे, बहुत ज़्यादा मैकेनिकल स्ट्रेंथ प्रोवाइड करेंगे. एडहेशन में भी हेल्प कर रहे हैं और मैकेनिकल स्ट्रेंथ भी प्रोवाइड करेंगे.